let's go over this. Very important that you understand this. If you understand this, then the chapter on genetics is going to be very easy for you.
What chapter is that?
Don't worry about it. Whatever it is, whatever it is. Just next chapter. I think that's next, or maybe chromosome is next chapter. Then chapter after that is the genetics chapter. But whatever. This you understand that then the material for genetics is going to be easy for you guys. Are you ready for it? That's why we took a break. We took a break because of this. I want to make you some sense a little bit. You got up, you stretched it a little bit. You should have got up and stretched. Anytime I give you guys a break, you should get up and stretch. You know, it will help you. Okay, so let's go over it. I don't know where I put my, here, click it. So you have a blue chromosome and a red chromosome inside of the cell. Normal, again, where this is occurring, this is in the ovaries of the females or testes of the males. Period. It does not happen in the brain cells. It does not happen in your biceps. It does not happen in little kid that is growing. The soccer player that cut her skin, his skin. You saw the video? The soccer player, not that. It is happening in the ovaries of the females and testes of the males. You have a blue chromosome, you have a red chromosome. Okay, am I making some sense? They go under the S phase of interphase, right? It becomes two sister chromatids. You have two blue, mine is green now. It's okay, they don't have green. I should have worn a blue shirt today, or a red shirt, but anyhow. So they have two of the chromosomes, right? You remember this from mitosis, similar to mitosis so far. And then the red becomes two also. Chromosome, 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 chromosome. Sister chromatid, sister chromatid. Sister chromatid, sister chromatid. Homologous chromosome. Right? Am I making some sense? Homologous chromosome. Same size, same length, same DNA material they have for inheritance, not DNA. They do not have the exact same DNA. Okay? So, what will happen during the first stage of meiosis, meiosis 1? In mitosis, you did not have mitosis 1, mitosis 2. You remember that? You did not have that. You just had mitosis, prophase, metaphase, anaphase, telophase. Okay, what was that acronym? Bring talk?
Please make a taco.
Please make a taco. I forgot what, I'm impolite, so I forgot about please. I said make a taco. I'm that type of person. I never tell my wife please, I say make a taco. Just like that. Make a taco. But I tell you guys, please make it. But anyhow, so meiosis 1, what will happen, the homologous chromosomes go to separate cells. This chromosome, the red, went here, and the blue went here. Do I make sense? And then in meiosis 2, which you will see it on Wednesday, meiosis 2, then the sister homologous chromosomes will separate. So, going back, in the cells of the ovary and testes, Reno and Maricela, is that right? Reno goes to one cell, that's what happened. Maricela goes to one cell. Am I making some sense, everybody? Then another cell division occurs. Reno, this arm goes to one cell, and this arm goes to another cell. Am I making some sense, everybody? And at the end, this arm goes to one cell, and this arm goes to one cell. At the end, when it's all over, they started out as one cell, right? How many cells they are incorporated? None of you and I open up your arm. You all went in separate cells, right? So at the end, how many cells is formed in meiosis? Count the cells. 4. 1, 2, 3, 4. At the end, we have 4 cells. Am I making some sense, everybody? But in mitosis, at the end, how many cells did you have? In mitosis, at the end, when it's all over, how many cells?
2.
2. Trying? Are you with us? You have 2 cells. So at the end, you have 4 cells. I hope I'm making some sense. Okay, so again, repeating again itself, the blue chromosomes multiply, becomes 2. Red chromosomes multiply, becomes 2 during the S phase of interphase. You remember that S phase? That happens during interphase. And then meiosis 1, the blue one separates, go to a different cell, and the red one separates, go to that one cell, and then this sister chromatids separate, comes to this cell. That sister chromatid separates, comes to that cell. This sister chromatid comes to that cell. That sister chromatid comes to that cell. Question from you. See if you're with me or not. The question is, can this one, or that one, or that one, or that one, can they undergo another cell division? Can they either go under mitosis or meiosis? The answer is yes or no. No. I wrote it big and bold here that after meiosis is over, they cannot undergo another division. It was bold. When you have sperm, sperm cannot divide, becomes another sperm. When you have egg, I'm lying a little bit in case of human, but don't worry about it. Those of you who had human sexuality or other courses, you know what I'm talking about. This cannot divide, become another egg. This cannot divide, become another egg. It's over. They're done. Am I making some sense a little bit? I'm lying in case of females, I'm lying a little bit, but that's okay. When the time arrives, we'll talk about it.
So, at the end of meiosis 2?
Yes, there's no more division.
But after meiosis 1, it divides.
Yes. After meiosis 1, of course. After meiosis, I didn't say that cell, I said these cells. Right? Am I making some sense? I said these cells, not these. Okay, let's go. Here we go, the whole 9 yard, the whole entire picture, meiosis 2, meiosis 1, the chromosomes become thickened, the chromosomes, they call it metaprophase 1. During lab practical exam, you must write down prophase 1, you must write down metaphase 1, you must write down anaphase 1, then telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. If you do not write down, you're looking at the slide of, today we will not look at the slide of meiosis. Today we'll look at the slide of mitosis. Am I making some sense? We'll look at the slide of mitosis. Whatever is related to mitosis, today we'll look at it in the lab. Meiosis is for next time. I hope I'm making some sense. And meiosis only occur in female's ovary and male's testes. That's it. Nowhere else. Nowhere else in the body. Nowhere else. Okay, let's go. So, division of in meiosis 1 occur in four phases. Meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and then of course cytokinesis. Prophase 1, in early prophase 1, each chromosome's pair with its homologous and crossing over occurs. Very, 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 very important concept, crossing over. I will ask quiz questions, lab questions, not lab questions, lecture questions from it. Crossing over. And what is crossing over? In a minute, hang on. I will let you know in a minute. Hang on. Okay? Hang on to it. X-shaped regions called chiasmata are sites of crossing over, crossover. Okay, meiosis 1 separates right here, Again, homologous chromosome lineup, kinetochore, same concept as those mitosis. I have nothing new to say, but make sure you look at it, you look at that slide and go over all of the labeling and make sure you know it. Meiosis, sure, let's look at it. I hope they're not.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber.
Prophase 1.
As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs.
Reno and
The close association of homologous chromosomes
Maricela.
allows segments of non-sister chromatids to trade places.
They're showing crossing over. Anyway, we'll talk about it.
This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell.
That's middle of the cell. Right here.
The next stage begins when homologous chromosomes separate from each other and move toward opposite poles.
Reno and
Each
Maricela
chromosome still
separated.
consists of 2 sister chromatids.
Okay, telophase 1.
This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes.
23 chromosomes, 23 chromosomes. Meiosis 2.
In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged
Half of
chromosomes
Reno and half
that
of
move to
Maricela.
opposite poles.
Half of
Nuclear
Maricela,
envelopes
half
reform,
of Reno.
and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay? Does it make sense, I hope? Meiosis and sperm formation. This is the real footage. It's not cartoon like last time, so it's not taking. This is the real one. Now you have 4 cells. 1, 2, 3, 4. It's supposed to be 4 cells in here. Okay, division 2 also occurs in 4 phases. Prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Meiosis 2 is very similar to mitosis. Okay, the fate. Why is it similar? Because of the fate of the chromosomes. You remember the chromosomes in mitosis, they separate the sister chromatids. You remember that? They separated, they went to separate cells. Same thing happens in meiosis 2. Sister chromatids are separating and they go to separate cells. I hope I'm making some sense. In meiosis 1, what happened, can you put your arms? In meiosis 1, Remember, in meiosis 1, we were together, and then we separated, right? I went to one cell, and she went to one cell. That's meiosis 1, right? In meiosis 2, am I making some sense a little bit? That's what happens in meiosis 2. So meiosis 2 is similar to mitosis models you're looking today.
Wait, and why is meiosis 1 not like mitosis? What makes them different?
What makes what difference between what?
Meiosis 1 and mitosis, why are they not similar?
Okay, great. Thank you. Please ask questions. If you don't ask questions, I don't know what you're. Okay, so let's go over. Good question. So you know about cancer, right? You all know what cancer. Hopefully you can answer a few quiz questions next time about cancer. Stick to your PowerPoints. Of course, if you don't understand the PowerPoints, read the textbook. That's what the textbook is. Okay, mitosis. I'm not going to draw all of the stages. Okay, is that okay with you? But right here, you have a cell, and you have a black chromosome and a red chromosome. Right? They duplicated. You know what I'm saying? They duplicated, and what happens in mitosis, you have, they line up in the middle of the cells, and the red chromosomes and black chromosomes line up in the middle of the cell, and then this cell will have one black and one red. This one will have one black and one red, right? And eventually, when this one wants to divide, this becomes 2, right? And this becomes 2, this becomes 2, this becomes 2, and they multiply again and again and again, right? This is we already agree. Now, in meiosis, you have the same thing, red and black, right? And then what will happen in meiosis, they line up in the middle of the cell. This is, meiosis 1. I should not confuse you guys. Meiosis 1, what will happen, this will go to one cell, and this will go to one cell. Meiosis 2, this becomes like this becomes like that. This becomes like this becomes like that. Okay, so what am I saying? This sister chromatid separate is the same as this sister chromatid separate. That's why here this homologous chromosomes separate. I went to a separate cells, non-wheat went to a separate cells. Am I making some sense? But the next stage, meiosis 2, half of me goes to one cell, half of me goes to another cell, just like mitosis. I hope I made sense. I hope I answered your question. Okay? Let's go. Then what? I need to talk about crossing over. Meiosis 2. So look at these diagrams at home. Go over them for yourself. Again, crossing over. Okay. Let's stop. I don't want to talk. Again, another very important concept, crossing over. You need it for the rest of your life. Crossing over is something important for the rest of your life. If you're going to biological sciences, if you're going to you're going to become a doctor, or you're going to become a physician assistant, whatever. Very important concept. Very, very important concept. Is there any question? Does anybody have any question? Okay, I'm going to return your lab.